I'm about to so
yeah.
Next one, kind of track that baby down.
Yeah, sure.
Clearly caught that ball.
Yeah.
This is fun. We're gonna get a little attention.
You guys ready to get started?
Yeah, Monday night football game.
I know that headline from
I know.
Let's go ahead and get the session started. A couple of announcements. Last week was the first session with a new system, so basically, for those on the outside, we learned a couple of things. I know a couple of you had some problems with the broadcast. The key thing is for everybody listening outside this room, first of all, we don't hear anything. It's muted on input into this room. So if there are any problems, I guess I should tell, any serious problems during the thing, call my cell phone, 849-0901, because that's really sort of the only way to interrupt us and let us know there's a broadcast problem. But the best thing is if everybody uses just their computer for audio and video, who's listening outside, there's no time delay. You'll get the audio and the video, and if you use a telephone separately from audio, there's a 30-second time delay between audio and video, which I imagine is very distressing if you're trying to follow this along. And the other problem with people using telephones outside is although all that audio is muted to us coming into the room, you weren't muted to one another. So people in Tacoma heard people in Everett with their dogs barking and their kids crying. But now, Drew tells me that should all be corrected. So those of you who are going to still use your telephone on the outside, hopefully you won't hear one another, but you're still going to suffer this 30-second time delay. So if we get to the point where nobody needs to use a telephone, it'll be ideal. It'll resolve all those problems. The other thing is, we no longer know with this system who's outside listening. It's not up on the screen here. So if you want to get CME credit, you have to send in the magic word of the day. And today's magic word is autumn. Autumn. There'll be a quiz later. All right, so today's speaker, I'm really honored to have Josh Boyce here from the Brigham in Harvard. Thank Merck for bringing him here. We'll be talking about aspirin exacerbated respiratory disease, new molecular insights into an old mystery. And I'll have him up here in just a second. Even his cryptic CV here is very impressive, just simply to say that he trained in pediatric pulmonology at the MGH and allergy immunology at the Brigham. He's currently the Sheffer Professor of Medicine in Allergic Diseases and Director of the Training Program at the Brigham. And I didn't realize how big the programs were back in Boston. He told me last night they had 9 fellows just at the Brigham alone, and if I got it right, another 4 at the MGH and others still at Children's. So a lot of people in our field being trained back in Boston. So it's something for us to emulate and for the fellows out there to be listening. So Josh, thanks for coming. It's all yours.
Thanks, Lad. Also thanks to Merck for flying me out here. Just to let you know that the blue shirt is emblematic of the political leanings of my state, and this one as well. So what I thought I'd do today is talk about aspirin exacerbated respiratory disease, which for somebody who is interested in lipid mediators, and you have some people in this room who are also interested in lipid mediators, I think this is an experiment of nature. It's certainly one of the most phenotypically distinctive allergic diseases. You almost can't mistake it for anything else. And because its phenotype is so consistent, there almost has to be an underlying mechanism that distinguishes it from all other asthma. And yet that mechanism or those mechanisms have remained evasive. So what I'm going to do is talk about some background and some history, but I'm also going to talk about some work that we've done in the last couple of years to try to understand the disease mechanism using a combination of approaches, translational approaches in humans with some approaches in animals as well. So I think since everybody here is clinically trained, you all know about the clinical entity, and I'm just going to highlight a couple of things to illustrate the real unknowns. So AARD most typically is a syndrome of adult onset asthma, rhinosinusitis, nasal polyposis, and the nasal polyposis is often the thing that drives these patients most crazy. And the diagnostic feature that distinguishes it from everything else is the respiratory reactions to aspirin and other drugs that inhibit COX-1. And that's very important because COX-2 selective antagonists do not provoke reactions. They rarely provoke reactions in these individuals. And this implies that there's something fundamentally different about the way that the cyclooxygenase enzymes function in this disease that is different from all other diseases, and we'll try to get a handle on that a little bit later. The pathology of AERD is marked eosinophilia of the sinonasal tissue and the lower respiratory tract, and on average, eosinophil densities are about 2 to 3-fold higher than individuals who have aspirin-tolerant disease. Biochemically, the hallmark is elevation of cysteinoleucotriene production as indicated by high levels of urinary leukotriene E4 levels at baseline, and especially after administration of COX-1 inhibitors. So during clinical reactions, you not only have a high baseline to start with, but when you give the COX-1 inhibitor, LTE4 in the urine skyrockets. And this implies, once again, that there's some sort of disorder in the way that cyclooxygenases regulate the function of and production of leukotrienes. So I'm going to try to answer 4 questions today. The first is, why are cysteinoleucotrienes overproduced in this disease? The second is, why is there so much eosinophilic inflammation in this disease? The third is, why don't all asthmatics have this disease? Why do only these people react to COX-1 inhibitors? And the last is, why does treatment with aspirin after desensitization improve disease control? Because that's a paradox, and it's one of the few things that we can actually do therapeutically for these people, and yet it's exactly the same drug that causes the reaction that can be used as a therapeutic once you desensitize them. So to understand the first part of this presentation, I need to review leukotriene production with you all. So cysteinoleucotrienes, hopefully you all know, are inflammatory mediators that are best known for their ability to constrict smooth muscle. And there are really 4 major cell types that have the entire complement of enzymes necessary to generate cysteinoleucotrienes from their own arachidonic acid. These are mast cells, macrophages, eosinophils, and basophils. The reason that they can carry out this function is they have the ability to take arachidonate and to metabolize it by this enzyme, 5-lipoxygenase, and create the precursor LTA4. And they also have the terminal enzyme leukotriene C4 synthase. And the job of leukotriene C4 synthase is to put reduced glutathione onto LTA4. And that's what creates LTC4, the first cysteinoleucotriene. Now, I've highlighted neutrophils up here and platelets down here in green for a very specific reason. Neither a neutrophil nor a platelet can make cysteinoleucotrienes. A neutrophil can make leukotriene B4 because it has an active 5-lipoxygenase enzyme. It makes tons of LTA4, and some of it gets hydrolyzed by a specific hydrolase enzyme to make LTB4. But a lot of the LTA4 that a neutrophil makes actually gets spilled out into the extracellular space because they make so much LTA4 that they can't convert it all to LTB4. And generally that LTA4 gets broken down very quickly into biologically inert metabolites. Platelets, on the other hand, can't make cysteinoleucotrienes because they don't have 5-LO, but they have a bucket load of LTC4 synthase. So what's that all about? It turns out that there are circumstances in cardiovascular inflammation, stroke, myocardial infarction, where you get platelets that stick to neutrophils in the periphery and in the tissues. And when that happens, the platelet becomes competent to convert the neutrophil's excess LTA4 into LTC4. Effectively, what that does is it creates a completely novel way for leukotriene production to get increased. And I'll show you why that's important in AERD, because we think that's a major contributor to this disease. So LTC4 gets moved to the extracellular space, very quickly gets converted to LTD4 by a specific gamma-glutamyl-leukotrienase. And LTD4 is the most powerful bronchoconstrictor known to exist in nature, over 1,000-fold more potent than histamine or methacholine. LTD4, though, has a very short half-life because there are extracellular dipeptidases that take the glycine residue off and create LTE4. And it is this molecule, LTE4, that is the only stable cysteinoleukotriene, the only one you can find reproducibly in urine and other biologic fluids, and consequently the one that is used in clinical studies to monitor cysteinoleukotriene generation. So about 12 years ago, Gilly Evans, who then was at Merck, cloned and characterized two receptors for the cysteinoleukotrienes that I think you're all familiar with, cis-LT1 and cis-LT2. And these two receptors, once they were identified, were characterized pharmacologically as receptors for LTC4 and LTD4, with LTD4 having high affinity for cis-LT1. Neither of these receptors bound LTE4 with any significant affinity. And we'll talk more about that in a moment, because everybody kind of forgot about LTE4 for about 20 years. But actually, LTE4, quite some time ago, when Dr. Christie was working with Tak Lee, was subjected to very rigorous analysis of physiologic effects in vivo in humans. And it's a powerful inflammatory mediator, and yet it has essentially no activity, these two receptors. So it must work by some other mechanism. So the therapeutics evolved pretty early on as people got excited about this pathway, with xyluton appearing in 1996. We use xyluton as a means of preventing severe reactions to aspirin when we want to desensitize somebody who's got really bad disease. It's generally not super popular for other purposes, but it works pretty well in that context, even though it's only a modest 5-lipoxygenase inhibitor. The Lucas drugs, which all block cis-LT1, have the ability to blunt bronchoconstriction in response to aspirin challenge, probably because they're interfering with this interaction of leukotriene D4 with a cis-LT1 receptor. And that validates that the leukotrienes are responsible for the bronchoconstriction occurring in response to provocation challenge. Again, we're going to talk a little bit about LTE4 here in a minute as a potential mediator that works through a pathway not dependent on the two receptors. Okay, so why are leukotrienes overproduced? I'll show you one potential reason. So these data come from a paper that was published in Blood about 4 months ago now by Tanya Laidlaw, who's one of our junior investigators. And Tanya got very interested in this issue of whether the platelet, with its LTC4 synthase enzyme, might be partly responsible for driving cysteinoleukotriene production. And in order to do that, a platelet has to adhere to a leukocyte that can make LTA4. And so this is a nasal polyp sectioned and stained with hematoxylin azure 2 eosin. And there's a sea of eosinophils and also some monocytes, some neutrophils. It's a heavily inflammatory focus. And here's the same polyp, double stained in green to identify the white cells with a CD45 common leukocyte antigen, and red, which is CD61, which identifies platelets. That's a very specific platelet marker. And there are tons of extravasated platelets, interestingly, in these nasal polyps. And on high magnification, it looks like those platelets are largely localized to the cell membrane of the CD45 positive leukocytes, suggesting these are platelet leukocyte aggregates in the tissue. And when the pathologist, who was blinded to the identities of the individuals, counted the numbers of platelet-adherent leukocytes and the fraction of the leukocytes that were adherent to platelets in polyps from people with AERD versus aspirin-tolerant polyps, there was a substantially higher number and higher percentage in the AERD samples. So, can you pick this up in the peripheral blood? And the answer is yes, very clearly. So these are measurements of platelet-adherent eosinophils, neutrophils, and monocytes expressed as a percentage of the total for each cell subpopulation using a fairly ingenious cytofluorographic strategy that Dr. Laidlaw worked out. And what she found was that when she compared the blood of individuals with AERD in black to aspirin-tolerant asthmatics in gray to non-asthmatic controls in white, there's a very substantial increase in the frequencies of platelet-adherent eosinophils, platelet-adherent neutrophils, and platelet-adherent monocytes in the peripheral blood. So these interactions are happening in the periphery as well as in the tissue. Notice that lymphocytes do not seem to have this pattern. It's really restricted to these three cell types, which are the three cell types that have the active 5-lipoxygenase enzyme and the potential to donate LTA4 to the platelet. So is that what is actually happening? So to get at the issue of whether the platelet was contributing to leukotriene production, we did some ex vivo analyses. And the first figure I'm showing you here is an LTC4 synthase activity assay. So in this assay, what you're doing is you're providing the cell with a substrate, LTA4, and asking that cell to convert the substrate to LTC4. That indicates how much LTC4 synthase is there. So when you look at isolated platelets from people with aspirin-tolerant asthma and compare them to those with AERD, there's no difference. They can both convert LTA4 to LTC4. When you look at the granulocytes, though, freshly isolated granulocytes, all comers here, look at the difference between AERD and aspirin-tolerant asthma. Tons of LTC4 synthase here, not as much here. And the question is, how much of this is due to adherent platelets? And to answer that question, we did a gentle trypsinization to remove the platelets, and we found about 60% of the LTC4 synthase activity in these granulocytes was actually due to platelets, and not to the granulocyte itself. And so, does that matter? So here are experiments where the cells are stimulated with calcium ionophore. In distinction to this assay where you're just feeding them LTA4, here you're asking them to make LTC4 from their own endogenous arachidonic acid. And once again, what you can see here is that for the AERDs, there's several-fold more LTC4 being generated here compared to the aspirin-tolerant granulocytes. And when you strip off the platelets, about 70% of that disappears. So this is largely driven by platelets. The question is, does that relate to the in vivo situation? And our best guess is that it does, based on these analyses. So here is urinary LTE4 in the three groups, non-asthmatic, aspirin-tolerant asthmatic, and AERD. This is baseline LTE4. This is very reproducible. Multiple groups have shown this. And here is the frequency of the platelet-adherent eosinophils as a percentage, platelet-adherent neutrophils, and platelet-adherent monocytes graphed versus the amount of urinary LTE4. And there's a nice positive correlation for all three subsets, suggesting that perhaps this is part of the way that you get hyperproduction of cysteinoleucotrienes in this disease, through this platelet-dependent transcellular pathway. So, to summarize the first part of this presentation, there's a markedly increased frequency of platelet-adherent leukocytes in AERD as opposed to aspirin-tolerant controls. There's a close relationship between those frequencies and urinary LTE4 levels. Platelet conversion of leukocyte-derived LTA4 to LTC4 is therefore a potential source of the excessive cysteinoleucotrienes that characterize this disease at baseline. And raises the question, if you had a means of preventing that interaction, would you have something that might work therapeutically in this disease?
This may be naive, but how do those two cells transfer the metabolite one to the other in the synthesis pathway? How do you get across the membranes?
Yeah, so LTA4 is actually quite membrane permeable. It's a highly lipophilic molecule, and as far as we know, there's no active transport that has to occur in order for that to happen.
So is the C4 being produced outside, extracellularly?
No, it's in the platelet.
Still inside the platelet.
It's in the platelet, yep. It's in the platelet. Okay. Now, to switch gears for a minute, I said we were going to talk a little bit about LTE4. LTE4 is the stable metabolite of this pathway, as I said, and therefore the most abundant cysteinoleucotriene due to its long half-life. And as I pointed out, it is commonly used as a marker for cysteinoleucotriene production in clinical studies. In AERD, LTE4 is a highly characteristic signature being elevated, as I said, in the urine at baseline and especially after challenge with COX inhibitors. And it's a very weak agonist at cis-LT1 or cis-LT2, has very trivial activity at those two receptors. And yet, we've known for some time, thanks to the work of Tak Lee and others, that LTE4 can promote bronchial eosinophilia when administered directly by in enterobronchial challenge or inhalation challenge, and also has this peculiar property of amplifying airways hyperresponsiveness to histamine in human subjects. And those are features that are generally not seen with LTD4. And interestingly, a paper by Tak Lee's group and Pandora Christie showed selective hyperresponsiveness to LTE4, but not LTC4 in individuals with AERD. And so these findings implied that LTE4 must be a mediator and not just a biomarker, and it must work by its own specific set of mechanisms. And this was before cis-LT1 or cis-LT2 had been cloned. Now, since pictures always are more illustrative than words, I'm showing you these data from a paper from the Hamilton, Ontario group of Paul O'Byrne, showing the results of inhalation challenges of mildly asthmatic individuals with LTD4 versus LTE4. And these patients were biopsied several hours after the inhalation challenge. And the biopsies showed that while LTD4 caused bronchoconstriction, it didn't promote the accumulation of submucosal granulocytes, but LTE4 did. Lots of granulocytes recruited to the airway in response to LTE4, and when these were enumerated, the granulocytes that really stuck out were eosinophils and metachromatic cells. There were also some increment in neutrophils, albeit more modest. So again, implication being LTE4 works by this pathway that amplifies mucosal eosinophilia, potentially very relevant to AERD, although these are not individuals with AERD. So about 4 years ago, when we were thinking about how we could understand the biology of this system a little bit better and understand why LTE4 was so pro-inflammatory and LTD4 didn't seem to be as much, we worked up a mouse system with the help of Sailija Paryachuri, who was then a postdoc in my group, who's now a faculty at University of Akron. And this was the system. It was basically sensitizing a mouse with ovalbumin and giving the mouse a really, really low dose of ovalbumin aerosol, just enough to cause a little bit of inflammation. And in this system, she administered either leukotriene D4 or leukotriene E4 down the airway about 30 minutes before each ovalbumin aerosol. And admittedly, this is a very contrived experiment, but it permits you to study the functions of LTD4 and LTE4 in isolation. And because it's a mouse, you can do the experiment in a critter that doesn't have cis-LT1 or 2, proving that LTE4 works by a separate pathway. And so these are experiments done in wild-type mice and receptor double knockout mice. So these are mice that have neither cis-LT1 nor cis-LT2. So if there are no other leukotriene receptors that exist, these mice shouldn't respond at all to LTE4. And the readout here is the percentage of eosinophils in the BAL fluid and the total numbers of eosinophils in the BAL fluid. The animals that received just low-dose ovalbumin are in gray, and the ones that got ovalbumin plus LTE4 are in black. And you see that LTE4 tremendously potentiates eosinophilia and does so just fine in the double knockout. So the double knockout has an LTE4 response that obviously can't be due to cis-LT1 and cis-LT2. And when we looked at histologic indices of inflammation, and goblet cell metaplasia, which is shown here in the PAS stain, which is a really nice and consistent feature of LTE4's effects in this animal, we got essentially the same results. The double knockout had an identical response to the wild type. So how is this working? And it's very complicated, so I'm not going to go into details, and I'm not going to talk about unpublished data, but what I will tell you is that there are at least two ways that LTE4 works. One is by a specific G protein coupled receptor, and the other one is by a very interesting and potentially therapeutically targetable pathway involving platelets. Now, I just got through telling you that platelets make leukotrienes. I'm now going to tell you that platelets are necessary for responses to leukotriene E4. And these are experiments done where we've depleted platelets from this mouse using an antibody strategy. This is obviously something you can't carry out in human subjects. Even the first-year fellows refuse to undergo platelet depletion. And you see the LTE4 gives us this nice goblet cell metaplasia here, and the accumulation of granulocytes around the airway. When we give anti-CD42, it disappears. And here are the quantitative data with the gray columns representing mice that were treated with an IgG antibody control. The black are the platelet-depleted mice. You see the whole LTE4 window disappears. And interestingly, you can also block this window with clopidogrel, which is Plavix, a drug that the cardiovascular docs use all the time. And not coincidentally, perhaps, Plavix is one of the few drugs that can inhibit the formation of platelet-leukocyte interactions. So we're very interested to see whether Plavix, we're actually not using Plavix, we're using Prasugrel, which is Effient, in a double-blind placebo-controlled trial. So we'll be able to answer the question whether, in fact, interfering with this P2Y12 axis is a useful therapeutic modality for individuals with this disease. So to summarize part 2, LTE4 is minimally active at cis-LT1 and cis-LT2, but it potentiates eosinophilia through platelets and P2Y12 receptors, raising the question as to whether P2Y12 antagonists might be useful in AERD. Okay, so I've talked about leukotrienes. I'm now going to flip gears and talk about the other major lipid pathway in this disease, which is the cyclooxygenase pathway. Has to be important because when you give cyclooxygenase 1 inhibitors, you get a reaction. And so the lead dog in the clubhouse in terms of relevance to this disease is prostaglandin E for reasons that I will explain. Prostaglandin E is an exceedingly abundant mediator. Every cell in your body can make it. And probably most of the time at baseline, cyclooxygenase 1 is responsible for generating some low level of prostaglandin E that serves a number of homeostatic functions. But with inflammatory diseases, there's a powerhouse system that gets induced to generate markedly increased quantities of prostaglandin E, and that is the cyclooxygenase 2 and microsomal PGE synthase 1 system. These two enzymes get co-induced in response to infection, in response to endotoxin, in response to a whole range of environmental danger signals. Consequently, almost every inflammatory disease will have some increase in prostaglandin E generation as part of the normal physiology. And prostaglandin E's effects are very complicated because of the fact that there are 4 receptors. So it's very diverse in terms of what it can do. And if you talk to a rheumatologist, they'll tell you prostaglandin E does bad things. It destroys bone, it causes pain, it causes swelling. It's one of the reasons why COX-2 inhibition works in rheumatologic diseases. But if you're talking to an allergist, prostaglandin E is actually anti-asthmatic and anti-inflammatory. And prostaglandin E's anti-asthmatic and anti-inflammatory properties may have something to do with the leukotriene pathway because one of the 4 G protein coupled receptors, the EP2 receptor, if stimulated, can send a signal that inhibits 5-lipoxygenase activation and inhibits leukotriene generation. So, is this at all relevant to AERD? So I'm going to show you a couple of pieces of data, not our data, but very elegant data that indicate that it might be. So this is from a very nice paper that appeared in a Japanese journal called Allergy International from a group of otorhinolaryngologists. The reason I like this paper is it's probably the only one that actually tried to directly measure the production of prostaglandin E in the target tissues. There's a lot of data on prostaglandin E metabolites in the urine, and there's actually no difference in urinary metabolites between AERD and aspirin-tolerant asthma. But in these studies, they're taking the sinonasal tissue from 4 groups. A control group who's undergoing head and neck surgery for other reasons, a non-asthmatic group undergoing sinus surgery, an aspirin-tolerant asthmatic group with polyps, and an AERD group. And here the tissue has the lipids extracted and measured, and you see there's this very sharp decrement in prostaglandin E levels in the nasal polyp tissue from people with AERD compared to the other groups. And not surprisingly, there's a corresponding increase in the cysteinoleucotrienes over here, as you might expect. So why is there this decrement in local production of prostaglandin E, and why is that important? So there have now been several studies reporting that in nasal polyps from people with AERD, there's a very, very low level, almost undetectable level of COX-2 messenger RNA relative to aspirin-tolerant asthmatic and normal sinus mucosa. So COX-2 expression may be impaired there. And in addition, the PGE synthase 1, the enzyme that pairs with COX-2, may also be impaired. And there was a lovely paper from Korea by Park et al. Showing that the PGE synthase gene in nasal polyps from people with AERD is hypermethylated. Hypermethylation is an epigenetic modification that can silence the expression of genes. And if you think about this, the implication here is if you don't have COX-2 and if you don't have the PGE synthase 1, this piddly little amount of PGE that's still there is probably all coming from COX-1. Probably all coming from COX-1. So if you give a COX-1 antagonist, what's going to happen? It's going to disappear. In addition, there are two lovely papers from Tackley and Chris Corrigan's group in JACI, one a nasal biopsy paper and the other a bronchial biopsy paper, showing very low levels of EP2 receptor expression in polyp and bronchus-associated leukocytes. Bear in mind, this is the receptor that can shut down the 5-lipoxygenase enzyme. So you've got three potential hits in this PGE pathway that may potentiate the activity of 5-LO and potentiate leukotriene production and set you up for a reaction. Well, that's all well and good hypothetically, but where's the proof that PGE is relevant to reactions? So this is about as close as we get. This is from a paper that's now about 15 years old by Sestini and colleagues in Italy. And what these investigators did is they took people with AERD, challenged them with aspirin on 2 occasions. On one occasion, they pre-treated them with prostaglandin E by inhalation. And on another occasion, they treated them with an inhaled placebo. And look at the difference. They completely blocked the aspirin-induced bronchoconstriction. Not only that, but they completely blocked the surge in urinary LTE4, suggesting that perhaps those two things are connected, that the actions of PGE in this disease may be to block that increment in leukotrienes that provokes the clinical reaction. So to turn for a moment from the human back to the mouse, I want to talk a little bit about a recent study where we tried to leverage the mouse again to answer a very specific question about a pathway relevant to this disease. And the hypothesis going in was that a deficiency of endogenous COX-2-derived PGE would potentiate airways inflammation and dysregulate the 5-LO pathway activity in response to allergen. Now, up until recently, really no evidence that you can get mice to have reactions to aspirin, so the best you can do is to try to recapitulate some of the inflammatory features of the disease. And so we used a model in which we induced pulmonary inflammation by administering an extract of house dust mite, DIR-F, which mice will respond to with repetitive administration by breaking mucosal tolerance and developing eosinophilic pulmonary inflammation. To get rid of COX-2-derived PGE, we used mice that lack microsomal PGE synthase 1. We didn't do a COX-2 knockout because a COX-2 knockout is going to be missing all sorts of prostaglandins, and we wanted to simplify the question and just focus on PGE. And so these mice were subjected to a protocol where they get 6 doses of dust mite allergen, very low dose, over a 17-day period, and then on day 18, the mice are euthanized for the studies. So what happens? So the first thing is that you really need this enzyme to ramp up prostaglandin E formation with pulmonary inflammation in this model. Here are the differences between the wild-type animal's lungs and the PGE synthase knockout's lungs in terms of prostaglandin E generation. There's about a 80 to 90% decrement. There's still some prostaglandin E, and again, that's probably coming from the COX-1 system, but it's very, very low. And when you look at what happens to their pulmonary eosinophilia, these are the BAL fluid cell counts. Here's the total cell number in the wild type and the PG synthase knockouts treated with dust mite. And here's the eosinophil number, and it's very, very striking. So the PG synthase, the absence of PG synthase markedly permits amplification of eosinophilia. And then when we look over here at the levels of leukotriene E4 recovered for the lung, there's about a 2.5 to 3-fold increase in the PG synthase knockout. So this begins to fulfill Koch's postulates that if you take away that inducible prostaglandin E, you develop hyperleukotrienism and you develop hypereosinophilia in the tissues. So, does this permit aspirin-induced bronchoconstriction? And so I'm showing you here one piece of data that will see the light of day fairly shortly, but is not yet published, in which the same exact experimental protocol was set up with the wild type and PG synthase knockout mice. But here, instead of sacrificing the mice and doing BAL fluid analyses and histology, what we did instead was to intubate and ventilate the mice and challenge them with lysine aspirin through the ventilator circuit. Lysine aspirin being a soluble form of aspirin that is used commonly in Europe to challenge patients and desensitize them. And we used lung resistance as our measure of aspirin-induced bronchoconstriction. And so these are the data, and they're very clean and very reproducible. So the PG synthase knockout mice, after they got their dust mite and were then challenged with lysine aspirin, bronchoconstrict, whereas the wild-type mice don't bronchoconstrict. So the absence of PGE synthase is the critical issue here. Not only that, but if you pre-treat the animal with montelukast, you actually block the lysine aspirin-induced bronchoconstriction. So this bronchoconstriction depends on the production of the cystinoLeukotrienes. We also now know from subsequent experiments that in this strain of mouse, if you just pre-treat the mouse with PGE, you also block the reaction. And if you treat the mouse with an EP2 agonist, you block the reaction. So this pathway is now beginning to be validated, that it is missing PGE, missing signal through the EP2 receptor, leading to hyperproduction of the cystinoLeukotrienes. And so to summarize this part, it appears that deficiencies in inducible PGE production can recapitulate aspirin sensitivity and dysregulate cystinoLeukotriene generation. Most likely this reflects loss of input through EP2 receptors. All right, so the last player I'm going to talk about here for a moment is thromboxane. So thromboxane is the major prostaglandin product of platelets. And thromboxane is generated primarily through COX-1. There is some production through COX-2, but COX-1 is the dominant player for this prostanoid. And once formed through thromboxane synthase, thromboxane elicits activation of cells through two receptors called TP alpha and TP beta. The TP receptors come from one gene, and the two isoforms come from alternative splicing. I mention that because if you delete that gene, you delete both receptors, as we'll see in a minute. Thromboxane, among the prostaglandins, is actually the most powerful bronchoconstrictor, more potent than prostaglandin D, and this is also an important mediator in the lung with inflammation because it can upregulate the expression of ICAM-1 on endothelial cells, and this being important to draw leukocytes into the tissue. So thromboxane very potently activates endothelial cells. Not surprisingly, given its production by platelets and COX-1, thromboxane is nicely suppressed by low-dose aspirin, and that's part of the basis by which low-dose aspirin elicits cardioprotection. And there's data in Japan that inhibitors of thromboxane synthesis and inhibitors of TP receptors can reduce sputum eosinophil counts and airways hyperresponsiveness, although modestly and not enough to get people excited about moving the drug forward. But the drugs are out there. Never been tried in AERD. So what is the role of thromboxane in AERD, if anything? Well, as I mentioned, since it's COX-1 dependent, it should get shut down when you do an aspirin challenge, and it is. And so these are data from Andrei Sheklik, who passed away this year. a really nice kinetic study in which individuals with AERD were challenged with aspirin, and 3 parameters are followed. Here's the drop in FEV1, which occurs very reproducibly somewhere between 2 and 4 hours, reaching its nadir. And the rise in urinary LTE4 shown here, peaking at about 4 to 6 hours and then dropping down. The thromboxane is here. This is measured out as a urinary metabolite. And you see it gradually declines, and it actually reaches its nadir around the time the lung function is recovering. So does that temporal relationship bear at all on a potential role for thromboxane in this disease process? So to show you one last set of experiments, again carried out in mice. And here the hypothesis being tested is that thromboxane, and probably platelets actually, are required for the pathologic changes caused by deficiencies in COX-2-derived PGE production. And so for this set of experiments, we're going back again to our prostaglandin E synthase deficient mice, and we're creating a double knockout by mating them with TP receptor knockout mice to create a mouse that can't respond to thromboxane and also doesn't make normal amounts of PGE. And so there are 4 strains of mice in these experiments: your wild type, your individual single knockouts, and your double knockout. Exactly the same protocol I showed you before with the dust mite, and exactly the same readouts I showed you before. So what happens if you take away the TP receptor from this mouse that has no PGE synthase? Well, once again, here's the high level of BAL fluid cellularity in the PGE synthase treated with dust mite, the high level of eosinophils. You take away the TP receptor, it's gone. You completely eliminate that window. That whole window of eosinophilia is dependent on the ability to signal through the TP receptor. Now, here is the piece of data that essentially everybody who reviews mouse airway papers demands that you do, and that is methacholine reactivity. And Teal and I were talking yesterday about how difficult it is to get a good window of methacholine reactivity in a black 6 mouse. But here you see that these dust mite challenged prostaglandin E synthase knockout mice have these gorgeous increase in lung resistance with methacholine challenge, so they're very hyper-responsive relative to every other strain. The double knockout is all the way down here. So you take away that TP receptor and they lose their hyper-responsiveness as well as their inflammation. So how does it work? Well, I mentioned a few moments ago that thromboxane is important for upregulating ICAM in the lung. And so we asked whether prostaglandin E might exert some sort of hierarchical control over that. And these are experiments where we looked at the inducible expression of ICAM on endothelial cells using a TP receptor selective agonist. And we got a nice increase in ICAM1, which wasn't a surprise. And when we treated these endothelial cells first with selective agonists of each of the 4 EP receptors, we found that the EP2 receptor agonist, the same one that shuts down aspirin-induced bronchoconstriction, was able to suppress the induction of ICAM1 by the TP agonist. And when we looked in vivo at the lung tissue, these are Western blots done on whole lung from these 4 strains used in this experiment, you see that the dust mite nicely upregulates ICAM1 protein in the wild type, more strongly in the PGE synthase knockout, and it's totally missing in the double knockout. So you take away TP, and in this model, you can no longer effectively upregulate ICAM. Okay, so summary number 4 is that deficient inducible PGE production amplifies the contributions of thromboxane and possibly platelets and TP receptors to the eosinophilia and the airway's hyperresponsiveness, suggesting that perhaps it is this disease in which a TP antagonist might actually have therapeutic benefit. Okay, so now what I want to do is take you through a set of summary slides to kind of wrap it all up and relate it contextually to AERD. So I think in aspirin-tolerant asthma, most likely what you have is a situation where you have a source of prostaglandin E coming from both COX-1 and 2, and you have signaling through the EP2 receptor on hematopoietic cells that have the 5-LO enzyme. And this signaling provides a brake against 5-lipoxygenase. So you'll get some leukotrienes forming both through the endogenous intracellular pathway. You'll also get them through platelets, but it won't be a high signal at baseline. The situation will be maintained in check. And then on the tissue side, in endothelial cells and maybe smooth muscle cells, you have exactly the same arrangement where the PGE is provided by the two enzymes. The EP2 receptor uses exactly the same inhibitory signaling to keep TP receptors in check and prevent upregulated expression of ICAM and thromboxane-dependent bronchoconstriction. So what about AERD? In AERD, you have two problems. The first is that the system that provides PGE through COX-2 is very deficient, rendering the prostaglandin E dependent on COX-1. And the EP2 receptor likely is also deficient. This essentially limits the extent to which this can check the 5-LO pathway. You're going to get a leak of leukotriene A4. You're going to get leaking through the endogenous leukotriene C4 synthase and through the platelet. And on the tissue side, this same arrangement is going to result in an impairment in the ability to control signaling through the TP receptor. You'll get more ICAM, you'll get more leukocyte extravasation as a consequence. And then when you give aspirin, you've eliminated that residual COX-1 dependent prostaglandin E. And at first, what you'll get is an explosion. You'll get a marked surge in leukotriene generation. You'll get all this LTD4 signaling through the cis-LT1 receptor to cause bronchoconstriction. But then, because you've shut off the production of thromboxane, you're going to, after some time, dampen down the inflammatory response, and the system will return to some sort of baseline in which you now have a therapeutic benefit from aspirin. There's almost certainly a bajillion things that high-dose aspirin does that are independent of this. I'm just raising this as one of the potential ways that a desensitization procedure might affect some efficacy. So I'm going to thank the people who really did the work here. Most notably Tanya Laidlaw, who's a really fun colleague. She does really good human work and is a draw for these patients. Tal Liu's a very talented PhD postdoc who did all the mouse work I showed you. And the mouse projects were actually initiated by two previous postdocs, Anders Lundqvist, who's back in Sweden with a faculty position, and Sailija Paryachuri did the LTE4 platelet stuff, and she's now at the University of Akron. I also want to thank Yoshi Kanaoka and Wei Zing for their contributions, and the NIH for funding the studies. Thanks.
I mean, this raises a number of potential clinical therapeutics, and you're obviously studying the prasugrel. The other one that seems obvious would be inhaled PGE2, and whether or not that's been looked at as a potential therapeutic.
So, a couple problems with, you're absolutely right. A couple problems with it. PGE2 is very irritating, and it makes you cough. So in the inhalation studies done by O'Byrne and others, it was very, very limiting. People didn't like that. That cough is probably receptor mediated, and there's nice work in the guinea pig to suggest that the EP3 receptor is responsible for the cough. So I think the proper therapeutic would be an EP2 agonist, with the caveat that if you're dealing with a person that has low EP2 receptor expression, would it be sufficiently efficacious? So, so your question is spot on, and one of the things that we're going to try to use this mouse system to study is to what extent we can bypass an EP2 defect through the other receptors, because at least one of them does have the ability to elicit a similar signal, and that's EP4. Please.
Do we know among aspirin-tolerant asthmatics more about why some respond really well to leukotriene modifiers and others it's like no effect?
No. No, I mean there are some studies that suggest that one can make such predictions based on genetics. And the genetics extend far beyond the genetics of CSL-T1. There are a number of polymorphic variants of other genes that apparently modify the response to CSL-T1 antagonists. There's very little replication data in the literature. These are hard studies to do. It's hard to get enough Ns. And there are some contradictions where a single nucleotide polymorphism that appears to be a predictor in one patient group doesn't seem to predict in another. So, simple answer to your question is no.
So, in clinic, trial and error is still the best way to...
Probably, yeah, probably. You know, I wonder, as I looked at what Teal showed me yesterday with their exercise-induced asthma studies and this data, the common thread is this ratio between cystinoLeukotrienes and prostaglandin E, which is higher in people with exercise-induced bronchoconstriction than asthma without exercise-induced bronchoconstriction. Clearly, way off the charts in people with AERD. You know, is that a predictor of Leucast responsiveness? If you had the ability to make that measurement in a standardized way, would that be a predictor?
I mean, there are actually studies in both. There's a study looking at aspirin-induced asthma showing a shift in the response to leukotriene modifier. And there's also a paper in children in which they did a randomized trial on children with exercise-induced bronchoconstriction and also showed a more consistent response to a leukotriene blocker than to an inhaled steroid in that particular population.
The other one, which I failed to mention, is Nathan Rabinowitz at Denver had a nice Jackie paper where he showed that the ratio between urinary LTE4 and exhaled nitric oxide was a predictor as well of responsiveness to the leukotriene receptor antagonists.
Since there's a close interplay between the Th2 cytokines and icosanoid formation, what aberrations have been shown in like the aspirin intolerant asthmatics?
Well, there is quantitative PCR data from nasal polyps showing higher levels of IL-5, which wouldn't shock anybody. There's a paper from Denver about a year ago now showing that IL-4 levels in the sputum are actually significantly higher in AERD than in aspirin-tolerant asthma. The question is that upstream or downstream? Very hard to answer. One of the interesting things about IL-4 and IL-13 signaling is that they are actually profoundly suppressive of COX-2 and microsomal PGE synthase expression and EP2 expression. So that whole cassette of molecules that are downregulated in AERD are targets of suppression by IL-4 and IL-13. So is that an IL-4, IL-13 signature? The argument against IL-4 and IL-13 driving the disease is, where's the IgE, right? Because those are class switch cytokines. These people don't have particularly high levels of IgE. It's a little higher than the non-atopic baseline, but it's nowhere close to what you get in allergic asthma.
Are any of these tests, for those of us not in an academic institution, useful at this point in separating out aspirin-sensitive versus non-sensitive asthmatic?
So we think the flow cytometry-based quantitation of platelet-adherent granulocytes probably is. We've seen essentially no overlap between the numbers that you see in the AERDs and the numbers that you see in the tolerant controls. What we don't know yet is specificity when you compare it across other diseases. So we're doing that now, and we actually have an application in for a patent, because we think it would be a nice diagnostic test. It's actually pretty easy to do once you have the antibodies, and the clinical lab could do it for you.
So urine leukotriene E4 is not...
Yeah, so urinary LTE4 in an asthmatic, it would be highly suggestive of AERD. But there are diseases in which you see sky-high levels of urinary LTE4. For example, eosinophilic pneumonias have very high levels of urinary LTE4. Hypereosinophilic syndrome has sky-high levels of urinary LTE4. Interestingly, those folks don't wheeze, generally speaking, the hypereosinophilic syndrome patients. They might have a little asthma, but for the most part, that's not the dominant pathology.
But theoretically, we would know that they didn't have those two before.
Theoretically, yeah. Theoretically, sure. Yeah.
So it could be used as a screen for people that have aspirin sensitive.
It could potentially. I mean, if you look at how the data kind of scatter out, there is some overlap between aspirin tolerant asthma and AERDs. There's no overlap after challenge. There, the signal gets huge. Actually, Pandora, do you want to comment on that?
Well, I remember this is some time ago that we did the study with the urinary leukotrienes, but there was an overlap, so you could have an aspirin tolerant patient who would have a high urinary leukotriene, but we never followed those patients up to see whether that was a marker that they might develop some intolerance.
And some years ago, Jeff Drazen had a paper in the Blue Journal where he showed that urinary LTE goes up during spontaneous exacerbations of asthma. And then, not AERD, just garden variety asthma off the street. And the levels drop during disease-free intervals, but we don't know what the kinetics are and how long it takes for it to drop to the baseline. So those are all caveats which would suggest that maybe it wouldn't be as specific as you'd like.
You sort of hinted at the two platelet agents, clopidogrel and Efient. They may have a role somehow clinically. Are you doing anything with them at this point?
Yeah, so we have, as part of our center grant from NIAID, we have a placebo-controlled trial of Efient. And the design is 4 weeks on Efient, 4 weeks on placebo with a crossover. And at the end of each of the 4 weeks, they undergo a challenge with aspirin. And the primary outcome variable will be the extent to which they experience symptoms with the challenge, as well as change in FEV1. And we're also looking at effects on all of these biomarkers, urinary LTE4, frequencies of platelet-adherent leukocytes in the periphery, exhaled nitric oxide, whole range of things. So yeah, I mean, we should be able to answer the question. I won't know what the answer is for another 3 years, but that's the plan.
Do you know what's driving the platelet leukocyte aggregation?
I would love to know that. So in the mouse, it looks like TP receptors are important.
Yeah.
In the human, when we've studied a handful of people after aspirin desensitization, we don't see a significant decrease, interestingly, in the frequencies of those aggregates, which would argue that maybe it's not thromboxane that drives it. There's no difference in P-selectin expression. There's no difference in PSGL1 expression on the leukocyte side. So the simple answers aren't the answers.
It's not obvious if there's platelet abnormality or a leukocyte abnormality.
So there is a very interesting old paper, a couple of them actually, from Monique Caprone way back when she was studying the platelets' role in cytotoxicity against schistosome larvae. And she, in collaboration with her husband and some people who were interested in AERD, actually collected about 40 people with AERD and about 15 controls with asthma, studied their platelets ex vivo. And interestingly, the platelet secretes something that kills larvae. And when they treated the platelets with aspirin ex vivo, only the AERD platelets released this something. So the readout was a cytotoxicity assay. They had no idea what the platelet was secreting, and they had no other readouts of platelet activation. So, we saw that paper. We thought, wow, maybe if we just add aspirin here, we can fire the platelet. And we looked at everything we could think of to look at that a platelet makes and that a platelet shows on its surface when it's activated. Nothing. So that doesn't mean that there isn't a platelet abnormality. We just can't find it yet.
For a disease that's so eosinophilic, you never mention any of the classical eosinophil attractants. Sort of following up on Bill's question, they just eotaxin and other chemokines, they're just not involved?
Been studied that no differences doesn't distinguish between AERD and aspirin tolerant asthma.
And how about BCAM? Does that play a role too?
Great question. Answer is we don't know. We tried to look by immunohistochemistry and the polyps and didn't get a good enough signal. Apparently VCAM is rather difficult to pick up histologically in tissues. We didn't see a difference at the transcript level. Thromboxane definitely upregulates VCAM on endothelial cells, just like the ICAM. I just didn't show you the data. So it's certainly possible, but we can't prove it yet.
When Mike Callender was here, he was telling us about putting Singulair directly onto the polyp through one of his nose spray concoctions. And can you do the same thing with any of the other leukotriene receptor antagonists? Or
I don't know, maybe.
is there direct toxicity? If you put them in tissue culture, is there any toxicity of those in these?
None that we can pick up at concentrations up to 10 micromolar, which is pretty high. Maybe you need a combination topical EP2 agonist and cis-LT1 antagonist. Maybe that's the ticket. I don't know.
Thanks very much for a great presentation.
Awesome.
For those listening outside, Drew may want to comment. I know we had audio problems along the way, and I think it still stems from having telephones.
Things sounded really good after the telephone.
Sorry to people on the phone if we spread them off.
So it appears we cannot use telephone audio.
I can't believe Art Bay didn't send me the question. Glenn, is it true that Art Bay didn't type in a question about this presentation?
What is true is Mike Weiss is the first one to contact me. Within 10 minutes, he told me the audio was ready. And then something